Let's continue with the renal physiology portions. So today we are going to learn about the concentration and dilution of urine. Learning objectives include describing the functions of antidiuretic hormone, describing the concentrations of tubular fluid as it passes through the different regions of the nephron, describe the mechanism of creating a hyperosmotic renal medulla, countercurrent multiplier system, then describing the countercurrent exchange system, vasa recta, and describing the disorders of renal concentrating or diluting ability in the next video set, or maybe in this video or next video, whatever it may be. So let's learn some major terms associated with this topic, such as osmolarity, measure of solute concentration defined as the number of osmoles. What is osmolarity? Measure of solute concentration defined as the number of osmoles of solutes per liter of solution. Osmolarity is the number of osmoles of solute per kilogram of solution. Then clearance, measurement of the renal excretion ability, and refers to a volume of fluid, usually blood, that is cleared and cleaned of the excreted substance per unit time. Then free water clearance, measurement of the renal ability to clear free water from the blood. It is an indicator of renal function and body water status. For the cells of the body to function properly, they must be bathed in extracellular fluid with a relatively constant concentration of electrolytes and other solutes. The total concentration of solutes in the extracellular fluid, and therefore the osmolarity, must also be precisely regulated to prevent the cells from shrinking or swelling. The osmolarity is determined by the amount of solute, mainly sodium chloride, divided by the volume of the extracellular fluid. Thus, to a greater extent, extracellular fluid osmolarity and sodium chloride concentration are regulated by the amount of extracellular water. The total body water is controlled by fluid intake, which is regulated by factors that determine this, and then renal water excretion, it is controlled by multiple factors that influence glomerular filtration and tubular reabsorption. In this chapter, we discuss the mechanisms that cause the kidney to eliminate excess water by excreting a dilute urine, and the mechanisms that causes the kidneys to conserve water by excreting a concentrated urine, the renal feedback mechanisms that control the extracellular fluid sodium concentration and osmolarity, then the thirst and the salt appetite mechanisms that determine the intakes of water and salt, which also help to control extracellular fluid volume, osmolarity, and sodium concentration. So antidiuretic hormone controls urine concentration. The body has a powerful feedback system for regulating plasma osmolarity and sodium concentration that operates by altering renal excretion of water independently of the rate of solute excretion. A primary indicator of this feedback is antidiuretic hormone, or ADH, also known as vasopressin. When osmolarity of the body fluids increase above normal, that the solutes in the body fluids become too concentrated, the posterior pituitary gland secretes more ADH, which increases the permeability of the distal tubules and collecting ducts to water. This mechanism increases water reabsorption and decreases urine volume, but does not markedly alter the rate of renal excretion of the solutes. When there is excess water in the body and extracellular fluid osmolarity is reduced, the secretion of ADH by the posterior pituitary decreases, thereby reducing the permeability of the distal tubule and collecting ducts to water, which causes increased amounts of more dilute urine to be excreted. Thus, the rate of ADH secretion determines to a great extent whether the kidney excretes dilute or concentrated urine, and this depends on the ability of the kidney to produce a dilute tubular fluid that is delivered to the distal tubes and collecting ducts where ADH acts. Then renal mechanisms for excreting dilute urine. That's the topic we are going to discuss. So when there is a large excess of water in the body, the kidney can excrete as much as 20 liters per day of dilute urine with a concentration as low as 50 osmoles per liter. The kidney performs this impressive feat by continuing to reabsorb solutes while failing to reabsorb large amount of water in the distal parts of the nephron, including the late distal tubule and the collecting ducts. approximate renal response in human after ingestion of 1 liter of water. Note that urine volume increases to about 6 times normal than within 45 minutes after the water has been drunk. However, the total amount of solute excreted remains relatively constant because the urine formed becomes very dilute and urine osmolarity decreases from 600 to about 100 osmoles per liter. Thus, after ingestion of excess water, the kidney rids the body of the excess water but does not excrete excess amounts of solutes. When the glomerular filtrate is initially formed, its osmolarity is about the same as that of plasma. To excrete excess water, it is necessary to dilute the filtrate as it passes along the tubule. This dilution is achieved by reabsorbing solutes to a greater extent than water, but this occurs only in certain segments of the tubular system as described in the following sections. So, tubular fluid remains isoosmotic in the proximal tubule. As fluid flows through the proximal tubule, solutes and water are reabsorbed in equal proportions, so little change in osmolarity occur. Thus, the proximal tubule fluid remains isoosmotic to the plasma with an osmolarity of about 300 mosmoles per liter. As fluid passes down the descending loop of Henle, water is reabsorbed by osmosis and the tubular fluid reaches equilibrium with the surrounding interstitial fluid of the renal medulla, which is very hypertonic, about 2 to 4 times the osmolarity of the original glomerular filtrate. Therefore, the tubular fluid becomes more concentrated as it flows into the inner medulla. So when we try to learn this mechanism, we can see next point, tubular fluid is diluted in the ascending loop of Henle. In the ascending limb of the loop of Henle, especially in the thick segment, sodium, potassium and chloride are widely reabsorbed. However, this portion of the tubular segment is impermeable to water even in the presence of large amount of ADH. Therefore, the tubular fluid becomes more dilute as it flows up the ascending loop of Henle into the early distal tubule, with the osmolarity decreasing progressively to about 100 mosmoles per liter by the time the fluid enters the early distal tubular segment. Thus, regardless of whether ADH is present or absent, fluid leaving the early distal tubular segment is hypoosmotic with an osmolarity of only about 1/3 the osmolarity of plasma. So, tubular fluid in a distal and collecting tubule is further diluted in the absence of ADH. And the dilute fluid in the early distal tubule passes into the late distal convoluted tubule, cortical collecting duct, and collecting duct. There is additional reabsorption of sodium chloride. In the presence of ADH, this portion of the tubule is also impermeable to water, and the additional reabsorption of solutes causes the tubular fluid to become even more dilute, decreasing its osmolarity to as low as 50 mosmoles per liter. The failure to reabsorb water and the continued reabsorption of solutes lead to a large volume of dilute urine. To summarize, the mechanism for forming dilute urine is to continue reabsorbing solutes into the distal segment of the tubular system while failing to reabsorb water. In healthy kidneys, fluid leaving the ascending loop of Henle and early distal tubule is always dilute, regardless of the level of ADH. In the absence of ADH, the urine is further diluted in the late distal tubule and collecting ducts, and a large volume of diluted urine is excreted. So, kidneys conserve water by excreting concentrated urine. So, the ability of the kidney to form urine more concentrated than plasma is essential for survival of mammals that live on land, including humans. Water is continuously lost from the body through various routes, including the lungs by evaporation into the expired air, and the gastrointestinal tract by way of the feces, and the skin through evaporation and perspiration, and the kidneys through excretion of urine. Fluid intake is required to match this loss, but the ability of the kidneys to form a small volume of concentrated urine minimizes the intake of fluid required to maintain homeostasis, a function that is especially important when water is in short supply. When there is a water deficit in the body, the kidneys form concentrated urine by continuing to excrete solutes while increasing water reabsorption and decreasing the volume of urine formed. The human kidney can produce a maximum urine concentration of 1200 to 1400 mSmols per liter, 4 to 5 times the osmolarity of plasma. Some desert animals, such as the Australian hopping mouse, can concentrate urine to as high as 10,000 mosmols per liter. This ability allows the mouse to survive in the desert without drinking water. Sufficient water can be obtained through the ingested food and water produced in the body by metabolism of the food. Animals adapted to freshwater environments usually have minimal urine concentrating ability. Like beavers, for example, can concentrate the urine only to about 500 mosmols per liter. Requirements for excreting a concentrated urine: high ADH levels and hyperosmotic renal medulla. So the basic requirement for forming a concentrated urine is 1. A high level of ADH, which increases the permeability of the distal tubules and collecting ducts to water, whereby allowing the tubular segments to avidly reabsorb water, and a high osmolarity of the renal medullary intestinal fluid. A high osmolarity of the renal medullary interstitial fluid, which provides the osmotic gradient necessary for water reabsorption to occur in the presence of high levels of ADH. The renal medullary interstitial surrounds the collecting ducts is normally hyperosmotic. So when ADH levels are high, water moves through the tubular membrane by osmosis into the renal interstitium, from where it is carried away by the vasa recta back into the blood. Thus, the urine concentrating ability is limited by the level of ADH and by the degree of hyperosmolarity of the renal medulla. We discuss the factors that control ADH secretion later, but for now it is the process by which renal medullary interstitial fluid becomes hyperosmotic. Which process? So this process involves the operation of the countercurrent mechanism. The countercurrent mechanism depends on the special anatomical arrangement of the loops of Henle and the vasa recta, the specialized peritubular capillaries of the renal medulla. In the human, about 25% of nephrons are juxtamedullary nephrons with loops of Henle and vasa recta that go deeply into the medulla before returning to the cortex. Some of the loops of Henle dip all the way to the tips of the renal papillae that project from the medulla into the renal pelvis, paralleling to the long loops of Henle are the vasa recta, which also loop down into the medulla before returning to the renal cortex. And finally, the collecting ducts, which carry urine through the hyperosmotic renal medulla before it is excreted, also play a critical role in the countercurrent mechanism. So countercurrent mechanism produces a hyperosmotic renal medullary interstitium. The countercurrent mechanism depends on the special anatomical rearrangement of the loops of Henle and vasa recta. So next, countercurrent mechanism produces a hyperosmotic renal medullary interstitium. The osmolarity of the interstitial fluid in almost all parts of the body is about 300 mosmols per liter, which is similar to the plasma osmolarity. The osmolarity of the interstitial fluid in the medulla of the kidney is much higher and may increase progressively to about 1200 to 1400 mosmols per liter in the pelvic tip of the medulla. This means that the renal medullary interstitium has accumulated solutes in great excess of water. Once the high solute concentration in the medulla is achieved, it is maintained by balanced inflow and outflow of solutes and water in the medulla. The major factors that contribute to the buildup of solute concentration into the renal medulla are the following: active transport of sodium ions and co-transport of potassium chloride and other ions out of the thick portion of the ascending limb of the loop of Henle into the medullary interstitium. So active transport of ions from the collecting ducts into the medullary interstitium then facilitate a diffusion of urea from the inner medullary collecting ducts into the medullary interstitium, and diffusion of only a small amount of water from the medullary tubule into the medullary interstitium, far less than the reabsorption of solutes into the medullary interstitium. So special characteristics of loop of Henle that causes solutes to be trapped in the renal medulla. The transport characteristics of the loop of Henle is very important, along with the properties of proximal tubules, distal tubules, cortical collecting tubules, and inner medullary collecting ducts. The most important cause of high osmolarity is active transport of sodium and co-transport of potassium chloride and other ions from the thick ascending loop of Henle into the interstitium. Because the thick ascending loop of Henle impermeable to water, the solutes pumped out are not followed by osmotic flow of water into the interstitium. Thus, the active transport of sodium and other ions out of the thick ascending loop adds solutes in excess of water to the renal medullary interstitium. This is some passive reabsorption of sodium chloride from the thin ascending limb of loop of Henle, which is also impermeable to water, adding further to the high solute concentration of renal medullary interstitium. So the descending limb of Henle's loop, in contrast to the ascending limb, is very permeable to water, and the tubular fluid osmolarity quickly becomes equal to the renal medullary osmolarity. Therefore, water diffuses out of the ascending limb of loop of Henle into the interstitium, and the tubular fluid osmolarity gradually rises as it flows towards the tip of loop of Henle. Step involved in causing hyperosmotic renal medullary interstitium. Keeping in mind these characteristics of the loop of Henle, let us now discuss how the renal medulla becomes hyperosmotic. First, assume that the loop of Henle is filled with fluid with a concentration of 300 mOs per liter, the same as that leaving the proximal tubule. Next, the active ion pump of the thick ascending limb of the loop of Henle reduces the concentration inside the tubule and raises the interstitial concentration. This pump establishes a 200 mOs per liter concentration gradient between the tubular fluid and the interstitial fluid. Step 3 is that the tubular fluid in the descending limb of the loop of Henle and the interstitial fluid quickly reach osmotic equilibrium because of osmosis of water out of the descending limb. The interstitial osmolarity is maintained at 400 mOs per liter because of continued transport of ions out of the thick ascending loop of Henle. Thus, by itself, the active transport of sodium chloride out of the thick ascending limb is capable of establishing only a 200 mOs per liter concentration gradient, much less than that achieved by the countercurrent system. Step 4 is additional flow of fluid into the loop of Henle from the proximal tubule, which causes the hyperosmotic fluid previously formed in the descending limb to flow into the ascending limb. Once this fluid is in the ascending limb, additional ions are pumped into the interstitium, with water remaining in the tubular fluid until a 200 mOs per liter osmotic gradient is established, with the interstitial fluid osmolarity rising to 500 mOs per liter. Then once again the fluid is in the descending limb reaches equilibrium with the hyperosmotic medullary interstitial fluid, and as the hyperosmotic tubular fluid from the descending limb of the loop of Henle flows into the ascending limb, still more solute is continuously pumped out of the tubule and deposited into the medullary interstitium. These steps are represented over and over with the net effect of adding more and more solute into the medulla in excess of water. With sufficient time, this process gradually traps solutes in the medulla and multiplies the concentration gradient established by the active pumping of ions out of the thick ascending loop of Henle, eventually raising the interstitial fluid osmolarity to 1200 to 1400 mOs per liter as given. Thus, the repetitive reabsorption of sodium chloride by the thick ascending loop of Henle and continued inflow of new sodium chloride from the proximal tubule into the loop of Henle is called countercurrent multiplier. The sodium chloride reabsorbed from the ascending loop of Henle, adding to the newly arrived sodium chloride, thus multiplying its concentration in the medullary interstitium. So, role of distal tubule and collecting ducts in excreting concentrated urine. When the tubular fluid leaves the loop of Henle and flows into the distal convoluted tubule in the renal cortex, the fluid is dilute with an osmolarity of about 100 m osmoles per liter. The early distal tubule further dilutes the tubular fluid because this segment, like the ascending loop of Henle, actively transports sodium chloride out of the tubule but is relatively impermeable to water. As fluid flows into the cortical collecting tubule, the amount of water reabsorbed is critically dependent on the plasma concentration of ADH. In the absence of ADH, this segment is almost impermeable to water and fails to reabsorb water, but continues to reabsorb solutes and further dilutes the urine when there is a high concentration of ADH. The cortical collecting tubule becomes highly permeable to water, so large amount of water are now reabsorbed from the tubule into the cortex interstitium, where it is swept away by the rapidly flowing peritubular capillaries. The fact that this large amount of water are reabsorbed into the cortex rather than into the renal medulla helps to preserve high medullary interstitial fluid osmolarity. As the tubular fluid flows along the medullary collecting ducts, there is a further water reabsorption from the tubular fluid into the interstitium, but the total amount of water is relatively small compared with that added to the cortex interstitium. The reabsorbed water is quickly carried away by the vasa recta into the venous blood. When high levels of ADH are present, the collecting ducts become permeable to water, so the fluid at the end of the collecting ducts has essentially the same osmolarity as the interstitial fluid of the renal medulla, about 1200 m osmoles per liter. Thus, by reabsorbing as much water as possible, the kidney from highly concentrated urine excreting normal amounts of solute in the urine while adding water back to the extracellular fluid and compensating for deficits of body water. Then urea contributes to hyperosmotic renal medullary interstitium and formation of concentrated urine. Thus far we have considered only the contribution of sodium chloride to the hyperosmotic renal medullary interstitium. However, urea contributes about 40 to 50% of osmolarity of the renal medullary interstitium when the kidney is forming maximally concentrated urine. Unlike sodium chloride, urea is passively reabsorbed from the tubule when there is a water deficit and the blood concentration of ADH is high. Large amount of urea are passively reabsorbed from the inner medullary collecting ducts into the interstitium. The mechanism for reabsorption of urea into the renal medulla is as follows. As water flows up the ascending loop of Henle and into the distal and cortical collecting tubule, little urea is reabsorbed because these segments are impermeable to urea. So, in the presence of high concentration of ADH, water is reabsorbed readily from the cortical collecting tubule and the urea concentration increases rapidly because urea is not very permanent in this part of tubule. As the tubular fluid flows into the inner medullary collecting ducts, still more water reabsorption takes place, causing a even higher concentration of urea in the fluid. This high concentration of urea in the tubular fluid of the inner medullary collecting duct causes urea to diffuse out of the tubule into the renal interstitial fluid. This diffusion is highly facilitated by specific urea transporters UTAI and UTA1. One of the urea transporters, UTA3, is activated by ADH, increasing transport of urea out of the inner medullary collecting duct even more when ADH levels are elevated. The simultaneous movement of water and urea out of the inner medullary collecting ducts maintains a high concentration of urea in the tubular fluid and eventually in the urine, even though urea is being reabsorbed. The fundamental role of urea in contributing to urine concentration ability is evidenced by the fact that people who ingest a high protein diet yielding large amount of urea as a nitrogenous waste product can concentrate their urine much better than people whose protein intake and urea production are low. Malnutrition is associated with a low urea concentration in the medullary interstitium and considerable impairment of urine concentrating ability. So, countercurrent mechanism and countercurrent exchange in the vasa recta preserves hyperosmolarity of the renal medulla. So, blood flow must be provided to the renal medulla to supply the metabolic needs of the cells in this part of kidney. Without a special medullary blood flow system, the solutes pumped into the renal medulla by the countercurrent multiplier system would be rapidly dissipated. There are two special features of the renal medullary blood flow that contribute to the preservation of high solute concentrations. Like the medullary blood flow is low, accounting for less than 5% of the total renal blood flow. This sluggish blood flow is sufficient to supply the metabolic needs of tissues that help to minimize solute loss from the medullary interstitium. So countercurrent exchange in the vasa recta preserves hyperosmolarity of the renal medulla. Blood flow must be provided to the renal medulla to supply the metabolic needs of the cells in this part of the kidney. Without a special medullary blood flow system, the solutes pumped into the renal medulla by the countercurrent multiplier system would be rapidly dissipated. There are two special features of the renal medullary blood flow that contribute to the preservation of the high solute concentrations. The medullary blood flow is low, accounting for less than 5% of the total renal blood flow. This sluggish blood flow is sufficient to supply the metabolic needs of the tissue that helps to minimize solute loss from the medullary interstitium. The vasa recta serves countercurrent exchanges, minimizing washout of solutes from the medullary interstitium. The countercurrent exchange mechanism operates as follows. Blood enters and leaves the medulla by way of the vasa recta at the boundary of the cortex and renal medulla. The vasa recta, like other capillaries, are highly permeable to solutes in the blood except for the plasma proteins. As blood descends into the medulla toward the papillae, it becomes progressively more concentrated, partly by solute entry from the interstitium and partly by loss of water into the interstitium. By the time the blood reaches the tips of vasa recta, it has a concentration of about 1200 mMol per liter, the same as that of the medullary interstitium. As blood ascends back towards the cortex, it becomes progressively less concentrated as solutes diffuse back into the medullary interstitium as water moves into the vasa recta. Although there are large amounts of fluid and solute exchange across the vasa recta, there is a little net dilution of the concentration of the interstitial fluid at each level of the renal medulla because of the U shape of the vasa recta capillaries, which act as countercurrent exchanges. Thus, the vasa recta do not create the medullary hyperosmolarity, but they do prevent it from being dissipated. The U-shaped structure of these vessels minimize loss of solute from the interstitium but does not prevent the bulk flow of fluid and solutes into the blood through the usual colloid osmotic and hydrostatic pressure that favor reabsorption of these capillaries. Under steady state conditions, the vasa recta carry away only as much solute and water as it is absorbed from the medullary tubule, and high concentration of solutes reabsorbed by the countercurrent mechanism is preserved. Summary of the urine concentrating mechanism and changes in osmolarity in different segments of the tubules. So, the changes in osmolarity and volume of the tubular fluid as it passes through the different parts of the nephron. Here we see it. So, first, proximal tubule. About 65% of the filtered electrolytes is reabsorbed in the proximal tubule. However, the proximal tubular membranes are highly permeable to water, so that whenever solutes are reabsorbed, water also diffuses through the tubular membrane by osmosis. Therefore, the osmolarity of the fluid remains about the same as the glomerular filtrate. Then, descending loop of Henry. As fluid flows down the descending loop of Henry, water is absorbed into the medulla. The descending limb is highly permeable to water but much less permeable to sodium chloride and urea. Therefore, the osmolarity of the fluid flowing through the descending loop gradually increases until it is nearly equal to that of the surrounding interstitial fluid, which is about 1200 mMol per liter, and the fluid blood concentration of ADH is high. When dilute urine is being formed, owing to low ADH concentrations, the medullary interstitial osmolarity is less than 1200 mMol per liter. Consequently, the descending loop tubular fluid osmolarity also becomes less concentrated. This is partly due to the fact that the less area is absorbed into the medullary interstitium from the collecting duct when ADH levels are low and the kidney is forming a large volume of dilute urine. Then thin ascending loop of Henle. The thin ascending loop is essentially impermeable to water but reabsorbs some sodium chloride. because of the high concentration of sodium chloride in the tubular fluid. Owing to water removal from the descending loop of Henle, there is some passive diffusion of sodium chloride from the thin ascending limb into the medullary interstitium. Thus, the tubular fluid becomes more dilute as the sodium chloride diffuses out of the tubule and the water remains in the tubule. Some of the urea absorbed into the medullary interstitium from the collecting ducts also diffuses into the ascending limb, thereby returning the urea into the tubular system and helping to prevent its washout from the renal medulla. This urea recycling is an additional mechanism that contributes to the hypoosmotic renal medulla. Then thick ascending loop of Henle. The thick part of the ascending loop of Henle is also virtually impermeable to water, but large amount of sodium chloride, potassium and other ions are actively transported from the tubule into the medullary interstitium. Therefore, fluid in the thick ascending limb of the loop of Henle becomes very dilute, falling to a concentration of about 100 mMol per liter. Then early distal tubule. The early distal tubule has properties similar to those of that thick ascending loop of Henle, so further dilution of the tubular fluid to about 50 mMol per liter occurs as solutes are reabsorbed while water remains in the tubule. Then late distal tubule and conical collecting tubules. In the late distal tubule and cortical collecting tubule, the osmolarity of the fluid depends on the level of ADH. With high levels of ADH, these tubules are highly permeable to water and significant amounts of water are reabsorbed. Urea, however, is not very permanent in this part of nephron, resulting in increased urea concentration as water is reabsorbed. This allows most of the urea delivered to the distal tubule and collecting tubule to pass into the inner medullary collecting ducts, from which is eventually reabsorbed or excreted in the urine. In the absence of ADH, little water is reabsorbed in the late distal tubule and cortical collecting tubule. Therefore, osmolarity decreases even further because of continued active reabsorption of ions from the segments. Then inner medullary collecting duct. The concentration of fluid in the inner medullary collecting duct also depends on 1 ADH and 2 surrounding medullary interstitium osmolarity established by the countercurrent mechanism. In the presence of large amount of ADH, these ducts are highly permeable to water, and when water diffuses from the tubule into the interstitial fluid until osmotic equilibrium is reached, with the tubular fluid having about the same concentration as the renal medullary interstitium, thus a small volume of concentrated urine is produced when ADH levels are high. Because water reabsorption increases urea concentration in the tubular fluid, and because the inner medullary collecting ducts have specific urea transporters that greatly facilitate diffusion, much of the highly concentrated urea in the ducts diffuse out of the tubular lumen into the medullary interstitium. This absorption of urea into the renal medulla contributes to the high osmolarity of the medullary interstitium and high concentrating ability of the kidney. So, several important points to consider may not be obvious from this discussion. First, although sodium chloride is one of the principal solutes that contribute to the hyperosmolarity of the medullary interstitium, the kidney can, when needed, excrete highly concentrated urine that contains little sodium chloride. The hyperosmolarity of the urine in these circumstances is due to the high concentrations of other solutes, especially of waste products such as urea. One condition in which this occurs is dehydration, accompanied by low sodium intake. Low sodium intake stimulates formation of the hormones angiotensin II and aldosterone, which together cause avid sodium reabsorption into the tubules while leaving the urea and other solutes to maintain the highly concentrated urine. Second, large quantities of the dilute urine can be excreted without increasing the excretion of sodium. This is accompanied by increasing ADH secretion, which reduces water reabsorption in the more distal tubular segment without significantly altering sodium reabsorption. And finally, there is an obligatory urine volume that is detected by the maximum concentrating ability of the kidney and the amount of solute that must be excreted. Therefore, if large amount of solute must be excreted, there must be accompanying by the minimum amount of water necessary to excrete them. So, obligatory urine volume. The maximum concentrating ability of the kidney detects how much urine volume must be excreted each day to rid the body of metabolic waste products and ions that are ingested. A normal 70 kg human must excrete about 600 mMol of solute each day. If maximal urine concentrating ability is 1200 osmoles per liter, the minimal volume of urine that must be excreted, called the obligatory urine volume, can be calculated as 600 osmoles per day is equal to 1200 osmoles per day is equal to 0.5 liter per day. This minimal loss of volume in the urine contributes to the dehydration, along with water loss from the skin, respiratory tract and gastrointestinal tract when water is not available to drink. The limited ability of the human kidney to concentrate the urine to only about 1200 osmoles per liter explains why severe dehydration occurs if one attempts to drink seawater. Sodium chloride concentration in the oceans averages about 3.0 to 3.4 with an osmolarity between about 1000 and 1200 osmoles per liter. Drinking 1 liter of seawater with a concentration of 1200 osmoles per liter would provide a total sodium chloride intake of 1200 osmoles. If maximal urine concentrating ability is 1200 osmol per liter, the amount of urine volume needed to excrete 1200 osmoles would be 1200 osmoles divided by 1200 osmoles per liter, or 1 liter. Why then does drinking seawater causes dehydration? The answer is that the kidney must also excrete other solutes, especially urea, which contribute about 600 osmoles per liter. When the urine is maximally concentrated, therefore the maximum concentration of sodium chloride that must be excreted by the kidney is about 600 osmoles per liter. Thus, for every liter of seawater drank, 1.5 liter of urine volume would be required to rid the body of 1200 osmoles of sodium chloride ingested in addition to 600 osmoles of other solutes such as urea. This would result in net fluid loss of 0.5 liter for every liter of seawater drunk, explaining the rapid dehydration that occurs in shipwreck victims who drink seawater. However, the shipwreck victims' pet Australian hopping mouse could drink with impurity or the seawater it wanted. Then urine specific gravity. Urine specific gravity is often used in clinical setting to provide a rapid estimate of urine solute concentration. The more concentrated the urine, the higher the urine specific gravity. In most cases, urine specific gravity increases linearity with the increasing urine osmolarity. So, urine specific gravity, however, is a measure of the weight of solutes in the given volume of urine and is therefore determined by the number of sizes of the solute molecule. In contrast, osmolarity is determined only by the number of solute molecules in a given volume. Urine specific gravity is generally expressed in gram per mole and in humans normally ranges from 1.002 to 1.028 gram per ml, rising by 0.001 for every 35 to 40 osmoles per liter increase in urine osmolarity. Quantifying renal urine concentration and dilution, free water and osmolar clearance. The process of concentrating or diluting the urine requires the kidneys to excrete water and solutes somewhat independently. When the urine is dilute, water is excreted in excess of solutes. Conversely, when the urine is concentrated, solutes are excreted in excess of water. The total clearance of solutes from the blood can be expressed as osmolar clearance. This is the volume of plasma cleared of solutes each minute, in the same way that clearance of a single substance is calculated, like C osmoles is equal to U osmoles into V by P osmoles. When U osmolarity is the urine osmolarity, V is the urine flow rate, and P osmolarity is the plasma osmolarity. For example, if plasma osmolarity is 300 mosmoles per liter, urine osmolarity is 600 mosmoles per liter, and urine flow rate is 1 ml per minute. The rate of osmolar excretion is 0.6 mosmoles per minute, and osmolar clearance is 0.6 mosmoles per minute divided by 300 mosmoles per liter, or 0.002 liter per minute. This means that 2 ml of plasma is being cleared of solute each minute. Then disorders of urinary concentrating ability. Impairment of the ability of the kidneys to concentrate or dilute the urine appropriately can occur with one or more of the following abnormalities, like inappropriate secretion of ADH. Either too much or too little ADH secretion results in abnormal water excretion by the kidneys. Then impairment of countercurrent mechanism. A hyperosmotic medullary interstitium is required for maximal urine concentrating ability. No matter how much ADH is present, maximal urine concentration is limited by the degree of hyperosmolarity of the medullary interstitium. Inability of the distal tubule, collecting tubule, and the collecting ducts to respond to ADH. Then failure to produce ADH, central diabetes insipidus. An inability to produce or release ADH with the posterior pituitary can be caused by head injuries or infections, or it can be congenital. Because the distal tubular segments cannot reabsorb water in the presence of ADH, this condition, called central diabetes insipidus, results in the formation of a large volume of dilute urine with urine volumes that can exceed 15 liters per day. The test mechanisms discussed earlier are activated when excessive water is lost from the body. Therefore, as long as the person drinks enough water, large decrease in body fluid water do not occur. The primary abnormality observed clinically in people with this condition is the large volume of dilute urine. However, if water intake is restricted, as can occur in the hospital setting when fluid intake is restricted by the patient is unconscious because of a head injury, severe dehydration can rapidly occur. The treatment for renal diabetes insipidus is administration of a synthetic analog of ADH like desmopressin, which acts selectively on V2 receptors to increase water permeability in the late distal and collecting tubules. Desmopressin can be given by injection as a nasal spray or orally, and it rapidly restores urine output towards normal. So, inability of the kidneys to respond to ADH. Nephrogenic diabetes insipidus. In circumstances, normal or elevated levels of ADH are present, but the renal tubular segments cannot respond appropriately. This condition is referred to as the nephrogenic diabetes insipidus because the abnormality resides in the kidneys. This abnormality can be due to either failure of the countercurrent mechanism to form a hyperosmotic renal medullary interstitium, or failure of the distal and collecting tubule and collecting duct to respond to ADH. In either case, large volumes of dilute urine are formed, which leads to cause dehydration unless fluid intake is increased by the same amount as urine volume is increased. Many types of renal diseases can impair the concentrating mechanism, especially those that damage the renal medulla. So also impairment of the function of the loop of Henle, as occurs with diuretics that inhibit electrolyte reabsorption by the segment, such as furosemide, can compromise urine concentrating ability. Furthermore, certain drugs such as lithium, used to treat manic depressive disorders, and tetracyclines, used as antibiotics, can impair the ability of distal nephron segments to respond to ADH. Nephrogenic diabetes insipidus can be distinguished from central diabetes insipidus by administration of desmopressin and synthetic analog of ADH. Lack of prompt decrease in urine volume and an increase in urine osmolarity within 2 hours after injection and desmopressin is strongly suggestive for morphogenic diabetes insipidus. The treatment for nephrogenic diabetes insipidus is to correct, if possible, the underlying renal disorder. The hypernatremia can also be attenuated by low sodium diet and administration of diuretic that enhances renal sodium excretion, such as a thiazide diuretic.